Dr. Voci is going to introduce our next speaker, which I'm super duper excited about. I was so excited when she said she would present today.
Thank you, Mindy. Yes.
Take it away.
Yes, thank you. Yes, we are all deeply honored to have our next guest with us. But Dr. Emmanuelle Waubant received her medical degree from the University of Lille in France. She then finished training in France as well as at the University of California, San Francisco in adult neurology, neuroimmunology, and MS clinical research. She is currently a professor of neurology and pediatrics at UCSF. She has served as the Race to Erase MS medical director since 2001. She also directs the Regional Pediatric MS Center at UCSF and is the president-elect of the Americas Committee on Treatment and Research in MS. Dr. Waubant serves or has served on numerous committees, including the Clinical Care Fellowship and Research Grant Review and Research Programs Advisory Committees of the National MS Society. She is chair of the Clinical Trial Task Force of the International Pediatric MS Study Group and a steering committee member of the U.S. Pediatric MS Network and International Pediatric MS Study Group. She directs translational research projects and mentors junior investigators on various clinical research and career development aspects in the field of MS and its related disorders. In 2018, Dr. Waubant also started an international consortium, International Women in MS, to help foster diversity and community spirit amongst clinicians and scientists from all over the world, as well as to promote diversity in professional organizations and help mentor junior women in the field of MS. Her specific interests include the translation of promising agents from the bench to bedside to help advance patient care, as well as risk factors for MS susceptibility and disease modifications in pediatric and adult MS. Dr. Waubant will be discussing the growing understanding of the microbiome as it relates to MS. So Dr. Waubant, we thank you for being part of our Yoga Moves MS community today. Thank you, and go ahead.
Thank you for the introduction, and I feel sorry to have a bio that is taking so many words. I should have shortened it.
No, you should be proud, never apologize.
I am not apologizing. It's just maybe not everything is needed in the bio. So today I will talk about some work that has been done all over the world. So you probably see my screen now. I want to give a critical update about what is known about gut microbiome and MS, because occasionally I've read either in scientific or lay articles things that were not really appropriate in terms of our current understanding and the translation to patient care. So the first thing I wanted to say is that why do we care about the microbiome? What's remarkable is that there's basically bacteria and viruses all over our body, inside and outside. There are tissues that are virtually sterile, but most of our skin and guts, for example, are covered in bacteria. That means that there's actually much more genes that are on our body than genes we host that are our own genes. So there's more than a 200-fold difference. So if we were just being genotyped, including all our skin and guts, most of what would be found would be actually not our own genes. The microbiome is not only made of bacteria, it's also made of fungi, some very small cell called protozoa, and viruses. They are, so the reason why people have focused on the gut microbiome is because it is really the largest bacterial community in our body. And it's contributing to digest some food and produces some vitamins that otherwise would not be absorbed well by the diet. And the barrier, so there's I'll show you afterwards on another slide, but there's a thin layer of cells that is a barrier between the bacteria that are in the gut and the entry in the gut. So it prevents the aggressive bacteria to actually enter the rest of our body. What's been very exciting is that over the past 20 years or so, there's been a growing understanding on how the microbiome and more of the work has focused on the gut microbiome, but there's also work that has looked at, for example, the dental or the sinus microbiome may regulate some of the immune response. This is a field that's reasonably new because the microbiome did not exist until the late 1990s, but there's been really a lot of enthusiasm about that developing field. The slide here is just confirming the role of the gut microbiome. So there are 100 trillion gut bacteria, so it's a huge community. Those bacteria typically contribute to the education of our immune cells, but they also can contribute to some diseases, including autoimmunity, we think, when there's an imbalance between different communities of bacteria. The imbalance of the bacteria in the gut can lead to a little bit of leakiness in the gut that allows the entry of some molecules that are in the gut and otherwise would not enter our body. And sometimes these are microbial products. The bacteria that live in the gut are actually modulated by all sorts of normal environments, but also less normal environments. For example, cigarette smoking or use of antibiotics are going to change in a major way, at least for several weeks, the gut microbiome. So on this slide here, I want to get a little more in depth into how we think the microbes in the gut may actually influence ultimately a disease like MS. So as I said, in the gut, there's a lining that is not a flat surface, but that is really very complex. And that lining is made of cells that protect from entry of bacteria or entry of different products of degradation from bacteria. And you can see that this lining is also very close to some nodes that are critical in the training of immune cells. And these little nest of immune cells are themselves in relation with lymph nodes around the gut that in turn contribute further to the training of immune cells. And then these immune cells can go in normal blood vessels. So we think that there's really the interplay between the normal bacteria that are in the gut, the integrity of the barrier of the gut, and what we refer to as mucosal immunity, because it's under the mucosal layer of the gut that may contribute to brain autoimmunity. Many years ago, so it was in the early 90s, I think that the work from John Government was really what started to open the eyes of researchers in the world of MS, because she published a paper that showed that mice that were raised in a completely sterile environment would not develop the animal model of MS called EAE. And when these mice were actually fed non-sterile food, they would ultimately be able to develop EAE. So these were mice that were engineered to develop EAE in the first place, but the contribution of the environment, whether it was sterile or not, was very clear. So it took more than a decade for people to get more excited about bugs, basically, in our environment and how they may influence animal models of MS, and then ultimately some interest in how they may be important in MS itself in humans. I selected only one of the many work that have been done in animal model, because I think that's been really the translation of what we know in humans back to the animal, and then suggesting that there was really a major role of the gut microbiome that occurred in MS. So this is a study that was very cleverly designed by some German colleagues that looked at stool samples from twin pairs. So they were similar twins, but one had MS and the other one was healthy. So they looked at those samples and how, if they were fed to mice, what was happening to these mice. And what you can see here on the slide is that for the healthy twin, there was actually few mice that over time developed MS. And these were mice that were also reasonably engineered to develop spontaneously EAE, the animal model of MS. But when this same colony of mice was fed samples that were coming from the twin that had MS, there was actually a majority of the mice that developed that animal model of MS, EAE. So that really raised a lot of hope in the community about what we could do for patients with MS. What we know from the gut microbiome is that there are changes in the different communities that are over time changing because of aging. And you can see here, it's just, I won't go in detail, but you can see that these are 4 or 5 of the largest families of bacteria that live in the gut. And you can see that the spread of these bacteria over time is a little different. There are the factors that influence also the profile of the bacteria that live in the gut. So I mentioned the age. We think that disease duration in MS may play a role, probably because of the medications that patients take. The geographic origin of patients, the season also makes the gut microbiome change, of course, the diet. And then lots of things such as supplements, vitamins, probiotics, and medications modulate the microbiome. And now we're starting to have a better understanding on how the genetic makeup of people influences the microbiome. And I wanted to lay a good picture of the landscape, because when you start looking at the MS studies, there's limitations that are in part related to these environmental changes that may affect the gut microbiome. So there's more studies that now have been published in MS, but overall, the vast majority of the studies have been small to moderate in size, which means they had between 10 and 60, 70 patients. And very often, at first, those studies enrolled patients who had very long disease duration and were on treatment for MS, which we know can affect the findings in the gut microbiome. Most of the studies have looked at patients who had MS from the perspective of looking at their stool samples. There was one study actually that looked at samples from the duodenal mucosa. So patients who were undergoing fibroscopy, for example, of their stomach because there were symptoms suggestive of maybe an ulcer. But most of the studies have looked at stool samples. When we look at the microbiome, we look at a lot of different ways to describe the microbiome. So we look at the overall community composition, and we call that diversity. So the diversity, how many bacteria are found in a given individual, or what's the number of bacteria that are found that differ between individuals. And then we can look also at the microbes themselves. So their presence or their abundance. And then we can look at pathways. So looking at large clusters of bacteria that like to hang out together, or that are involved in specific metabolism. Going back to the German study I mentioned earlier, I think that study and then subsequent studies have really showed that the diversity was not very different from a healthy twin and their other twin that had MS. And I think that's basically been reproduced most of the time by other studies. So if it's not the overall diversity, what is it? Is there specific bacteria that are actually associated with MS? Well, the response to that is very complicated because, as I said, most of the studies were small. They may have been done in patients who had MS for a very long time, that had been on different types of treatment that can alter the findings. And then the controls that are used in these studies to find differences between MS patients and people who don't have MS, the controls may also have been sometimes a little heterogeneous in the way they were selected. So here it's just a slide showing you some of our findings in a pediatric MS study, looking very early in MS, after MS onset. And the slide is just recapitulating here some of the bacteria that are more abundant in patients with MS compared to controls. And then you see also there's 15 or so bacteria that are less abundant in patients with MS compared to controls. And some of these bacteria are actually specific to our study, but some have been reported by other people who have looked at differences between adult MS and adult controls. And the reason I was interested in pediatric MS is because we know that children have been exposed to much less environmental changes because their life has not been as long. And as such, the findings may be less polluted, if you will, by different exposures to various medications or diet changes and so on. Then what we can do when we look at the microbiome, we can look at what's the function of the bacteria that are found increase or decrease. And here, this was a study that looked at metabolic pathways. So, for example, a group of bacteria that together, for example, here you see this is a large group of bacteria that contribute to the making of urate, for example. And so in that pathway, there's a lot of bacteria, but there's another pathway that was underrepresented also in MS patients that had only 3 types of bacteria contributed to the pathway. So we can look at bigger pictures at just one bacteria alone, because we know that there's a fair amount of redundancy with certain bacteria, and some can contribute similarly to the metabolism. Then some studies have looked at how the microbial abundance in the gut may influence the expression of genes that regulate the immune response. And so I will not go through this, but I just wanted you to know that we can also look at increase or decrease in the stimulation of some immune cells relating to the abundance or the presence of specific bacteria in the gut. And these immune cells are actually in the peripheral blood, showing really that what's happening in the gut has consequences that are detectable at the level of the blood in patients. I'll switch quickly here to something that in my mind is even more valuable to discuss with patients who have MS. So I was just talking about differences between patients who have MS versus patients who don't have MS, and how we think these differences may influence the onset of MS. But now I'm going to talk about changes that may influence the course of MS. So, for example, taking patients early on who have MS shortly after their disease onset, and then monitoring over time how the abundance of specific bacteria may be associated with increased risk of relapse or scars on the MRI. So this is a slide we often use to talk about disease activity in MS. So we can talk about the clinical aspect of the disease for patients who have relapsing MS. We can see bouts of symptoms that we call relapses that recover or don't recover fully, with over time a secondary progressive phase. But we can look also under the radar screen at what happens at the MRI level. And we can see here, for example, every time there's a yellow arrow, basically new scars that develop. And we know that there's more scars that develop on the MRI than flares of MS that patients would experience. So keeping that in mind, we designed a study in pediatric MS, again, because we think that there's less kind of changes that are related to, for example, having high blood pressure or smoking, and that we're closer to the reality of what happens in our body. So we enrolled 55 pediatric onset MS cases throughout the US. And we actually identified controls who were healthy in the same age range. We collected a sample shortly after onset of their disease, and then we looked at what happened afterwards in terms of their clinical symptoms, but also their MRI changes. The study identified that there were two types of bacteria that may be associated with an increased risk of relapse, so an increase in the abundance of these two bacteria, Blautia and Christenacell. So some of these bacteria, as a side comment, have very complicated names. That's why I'm trying to avoid mentioning them, because I think it's all a big... I wanted to have a big picture of what's happening in the field. So here in our study, we found two bacteria that, if they were increased in abundance, those patients tended to have more relapses. And similarly, we had also one bacteria that was associated with presence of new lesions on the MRI. But that bacteria, actually, an increase in the abundance of that bacteria was associated with a lesser chance of having new scars develop on the MRI. So then we looked at communities of bacteria. As I said, bacteria like to hang out with some of their little bacteria friends. So we're really more and more looking at communities in general, as opposed to a single bacteria. And here in this same study, we identified two clusters of bacteria that were strongly associated with the presence of new lesions on the MRI, whether we looked at those that were enhancing or those that were not enhancing. So that was very interesting. However, I think at the end of the day, what do we know and what is going on in terms of translating all this knowledge for the care of patients? I would say that at this point, there's really limited amount of information about how the gut microbes or clusters of those microbes are associated with the risk of MS flare or new scars on the MRI. So there's very little so far to guide intervention. Nevertheless, there's different researchers who have jumped ahead and have tried to do some intervention studies. So try to treat patients with MS with different ways to intervene on the microbiome. There was a small study of probiotic supplementation that was done at Harvard that just was that intended to look at the changes in the immune response, but not at all at the efficacy because the study is way too small. Also, I want to say that there's hundreds of different probiotics. So which one would you pick? And in that case, the researcher picked a probiotic that was offered by them with a grant to study in MS. Some people think also that, for example, prebiotics such as dietary fibers could be an interesting way to modulate the microbiome. Live biotherapeutic products also are considered. Some people have started to do microbiome transplants based on the animal model that I discussed early in my presentation. I think that the transplant, in my mind, is not always necessarily a sustainable way to do it, because it probably will take long treatment to alter sustainably the gut microbiome. So just do one transplant probably will not lead to improvement of a disease such as MS. So in conclusion, I wanted to say that we still need to establish causality between those bacteria and the disease course. It's really not completely clear whether there's a true gut microbiota signature for MS risk, and also how these bacteria contribute to MS course. It's really important to sort that out so we can design trials to intervene based on evidence that will make us less likely to fail. So I wanted to thank you for your attention, and happy to take questions.
Thank you so much, Dr. Waubant. Oops, there's my video. So we have a few questions. One is, do you recommend a specific diet or what not to eat to help with gut health? Which is a segue to our next speaker, but perhaps you could answer it briefly.
So I think I will let the next speaker touch on that, because I think when we talk about diet, it's not only diet for MS, but it's diet also for general health. And diet, as it may provide nutrients that are good, or nutrient, for either training the immune response or not. So I will let and modifying the microbiome. So I think the answer, I don't want to steal any thunder from the next speaker. So I will not answer that question. Sorry.
That's okay. This is all good transition to our next speaker. Next question, this may also be a prepper for Dr. Katzen. Let's see, has healthy gut been defined, or does it depend on each person?
So healthy gut, I don't think it's really clearly defined, but as long as there's no leakiness of the gut, it means that the gut microbiome is healthy. I think when there's something called dysbiosis, which is basically an imbalance between different communities that result in inflammation and breakdown of the gut barrier, then that's when it's not healthy anymore. There's also instances of unhealthy microbiome that is related to kind of increased abundance of bacteria that may be really very aggressive bacteria that usually are not hosted in the gut. That can result in very severe infection, and that type of complication is typically coming after antibiotics, a long and aggressive antibiotic course. This is a rare complication of antibiotic course, but that's when people have started to develop transient microbiome transplant, because in that situation, we know exactly which bacteria we're trying to fight and get rid of, and which bacteria actually help to do that. So, I think that there's a lot of variability from one individual to the other. There's millions of people who have a healthy gut microbiome, but there can be huge differences depending on their type of diet, the season, because the season influences the diet, and so on and so forth. So pretty complex stuff.
It sure is. And I'll let you determine whether we should let Dr. Katzen answer them, or whether you're going to. they keep moving the questions on me. When someone has MS, can their microbiome be studied to give baseline today?
Yeah, so that's a good question, because very often what has happened is that the microbiome studies, because of the complexity to set them up and also the cost, have typically collected only one sample per patient. Now there's more and more attempts to try to collect several samples from a patient over time to understand really the variability and the consistency across the microbiome and how it may relate to MS. So people can give samples, but typically you need to work with a group that is doing research on that topic. So very often we actually freeze samples before we analyze them, because we need to have enough to analyze. And those samples are stable at -80 degrees in a freezer.
Do you recommend any probiotics for gut health?
So I don't, because there's so many probiotics, and that's a question many of my patients ask. I think that a very diverse diet is probably with lots of fruits and vegetables is probably contributing to a healthier gut microbiome. In some instances, I recommend probiotics to my patients if they have recurring urinary tract infections, because the urologists are starting to use more and more probiotics to kind of rebalance a little bit better the microbiome, the gut microbiome, but also probably the microbiome around the pelvic floor.
Do you think kefir, kombucha, or yogurt can be beneficial for gut health?
Probably. There's no direct, there are some studies in MS. I don't know of a study that has looked at that, but this is definitely something we're capturing when patients donate a stool sample. We're asking whether they take some of these products, because it can change the findings.
Is gastritis a symptom of dysbiosis?
Gastritis it can be a symptom of lots of different things. It can be related of inflammation of the stomach because of medications that people take that can, like non-steroidal anti-inflammatories, can irritate the lining of the stomach. Some people have also increased stomach juices that can do that. And sometimes it can be related to dysbiosis. For example, so it's not gastritis, but it's been known for a long time now that, for example, stomach ulcers, some were actually associated with Helicobacter. So some bacteria can induce stomach ulcers. And what's actually very tricky is that we think in some conditions, some bacteria may participate to increase the risk of some cancers, for example. But at the time the cancer is diagnosed, if you look at these bacteria, they're not there anymore. So, the biology is very complex, and it's important to understand the limitations of what we do to be able to really interpret appropriately the findings of our studies.
Okay.
I have a question about, is C. diff common with MS patients?
Is C. diff common with MS patients?
No, and C. diff is actually the bacteria I was referring earlier as a very aggressive bacteria that has been treated with microbiome transplants. Some people have C. diff and don't have symptoms of it, but some people have very severe gut complications. But it's not necessarily common, more common in MS.
Does the healthy gut in MS patients change with COVID-19 vaccines?
You guys have really complicated questions. I don't think people have looked at that. So we know that different medications for MS, for example, oral treatments or even the injectable treatments or the IV drip treatments are associated with changes in the gut microbiome. I don't think there's really data looking at what happens after a vaccine. But one has to understand that when there's interventions such as taking antibiotics or steroids for an MS relapse, the changes that are induced by this treatment in the gut microbiome are actually temporary. And typically, the gut communities go back to previous to the treatment within a month or 2 months. It's only with treatment that you take on a regular basis long-term that can induce long-term changes in the gut microbiome. So I would not expect a vaccine induces long-term changes in the microbiome.
So there's one, I think she's clarifying that imbalance in the gut impacts inflammation. So are you saying that an imbalance in the gut impacts inflammation?
Yeah, so we know that there are differences across people and within the same person across different seasons or diets that don't result in inflammation, but there are some imbalances in which some communities become predominant or become so tiny that it's promoting inflammation in the gut. So we know that some diseases like Crohn's disease and irritable bowel syndrome, for example, can be associated with some of that. But there's also more and more evidence that some of these imbalances can result in different type of autoimmune, whether it's lupus or MS.
Okay, how does someone know if they have leaky gut? That's going to be your last question.
We don't test for that because it's really, we don't have any good way to evaluate that. You could look at the composition in protein in some of the stool samples, or look at the immunoglobulin level of different types of immunoglobulin, but this is not something that's done commercially. And I don't think that in the field of MS, there's any reason to test for that at this point, because we don't have an answer of, is it bad for everyone or it doesn't matter, it's just there.
Thank you so much, Dr. Waubant. That was, I've been looking forward to this all year since you said yes.
Wow.
It's just such a specialty and it's such new information. So thank you so much.
Well, my pleasure, and congratulations for a great meeting and a great lineup of talks.
Yeah, we're very fortunate.